So today in this lecture, we are going to discuss the very basics of molecular biology. So here you can see what is life. Molecular biology forms the basis of your life. Okay, moving further. Hi. So how is molecular biology different from biochemistry? Molecular biology is a part of biochemistry and is the study of nucleic acids and proteins, and also the interactions or reactions occurring between the nucleic acid and associated macromolecules. It completely revolves around the central dogma of biology, while biochemistry is mainly concerned towards the molecular genetics, metabolism, synthesis, and biochemical processes studies of all the biomolecules. And a takeaway message from these two subjects are that molecular biology is a marriage of biochemistry with genetics. So the birth of genetics, the father of genetics is, as we all know, Gregor Mendel. But after all this discovery, we come to know about various terminologies like chromosomes, DNA, genes, allele, genome, etc. So I'll be clearing these all in the next slide. So here is the basic diagram of DNA versus genes versus chromosome. As you can see, that here is a cell, nucleus. Nucleus consists of chromosome. As you might have read in your previous classes that haploid chromosomes, somatic cells consist of a diploid set of chromosomes, while the gamete consists of haploid set of chromosomes. So chromosomes are nothing but DNA supercoiled with histone. You can see this. As we go on unwinding that chromosome, we can see DNA being coiled with histone proteins. Okay, and the gene is nothing but a segment of DNA, and DNA is a polypeptide, polynucleotide. Sorry, I'm sorry to use that word polypeptide. It is a polymer of nucleotide, that is polynucleotide, consisting of a sugar backbone. You may see this structure, pentagon-like structure is nothing but ribose. Okay, and these are your nucleotide base pairs. Okay, so this is how DNA is compactly packed in a cell. As you may see here, all the definitions, it's clear. I've already made them very clear. Okay. So here's a brief history of the events and the scientists who have contributed towards the discovery of genetic material and in the entire genome, and it is all these discoveries that form the basis of further applications of biotechnology, and it also helps in pharmacogenomics. Pharmacogenomics is nothing but customized medicines as per the genetics of a population. Okay, so in search for genetic materials, a various number of studies have been conducted, and the most importantly being the transforming principle, the discovery of transforming principle by Griffith, and the further validation by Sia and Dawson. Avery, Macleod, and McCarty validated it even more by saying that it can be possibly DNA, with the final confirmation from Hershey and Chase that yes, DNA is the one. Okay. So one by one in the next slides, we'll be studying about all these various experiments in detail. So you see, the Griffith's experiment was a stepping stone for the discovery of genetic material, and he conducted all his experiments with Streptococcus pneumonia. Okay. So during the experiment, Griffith cultured the S. pneumonia bacteria, and which showed two types, two strains. One is the R strain, one is the S strain. R means rough colonies and it is non-virulent. And S is the smooth colony because it consists of a capsule made of mucus coat and it is virulent. Okay. So Griffith injected both S and R strains into the mice. The one which was infected with the S strain developed pneumonia and died, while the one who was injected with the R strain was alive. In the second stage, Griffith killed the S strain bacteria. Heat killed. It was heat killed, remember, and injected it into mice, but still it was alive, like we did in the case of live R strain. But here comes the last set of experiment in which he mixed in which the broth consisted of heat killed S strain plus living R strain, which were injected into the live mice. But to his surprise, the mice died. In addition, he found the living S strain in the sample recovered from dead mice. So to conclude, based on the observation, Griffith concluded that R strain bacteria could have been transformed by S strain bacteria. That is, the R strain inherited some transforming principle from the heat killed S strain bacteria, which made them virulent. And he assumed that this transforming principle is the genetic material, but it is still not concluded that it is DNA only or not. But there is one more thing, that is it due to the mutation in bacteria? There were some doubts, like it is due to mutation of bacteria, or is it due to the living host which is making some changes and letting it succumb to pneumonia? No. It will be further validated by Sia and Dawson. Okay. So Sia and Dawson in 1931 again performed the same experiment, but in vitro, just to show that mice had played no role in the transformation process. You can see in the diagram that when heat killed S strain and living R strain, when they were incubated together, the agar plate consisted of S strain colonies. So mice had literally no role in it. And there was no mutation as well. But it is still a doubtful thing that whether it is protein or DNA or RNA, what is still that genetic material? We are still stuck in the term transformation principle. We still don't know what is exactly that genetic material. So what genetic material is being transformed or being transferred into the next organism? The further two groups which I will be talking about have validated these experiments just to conclude that yes, DNA is the one. Okay, now the next group comes Avery, Macleod and McCarty, that the transforming principle is DNA. Okay, so it was a turning point in the discovery of genetic material. However, it failed to explain the biochemistry of that material. Okay. Avery and his team extracted and purified proteins, DNA, RNA and other biomolecules from the heat-killed S strain, and they selected the DNA from heat-killed S strain. Now they had 4 sets of experiments. That is, one consisting of DNA of S strain, R strain and then they incubated it, and there were S colonies, S strain colonies. Now, in the next set of experiments, this broth was subjected to treatment with protease extracted, which is present in trypsin or chymotrypsin. Trypsin and chymotrypsin are protease enzymes which cleave the protein. And again, the observation was there was growth of S strain. Even when the broth was subjected to treatment with RNase enzyme, again the observation was the growth of S strain colonies in the agar plate. But when the broth was by broth I mean to say is the DNA from S strain plus the R strain bacteria. But when this broth was subjected to treatment with DNAs, then there were no colonies. There was no growth because the DNAs cleaves the DNA from S strain, and here you can see it's the serum that precipitates pure R strain. That means it is the serum consists of those enzymes which precipitates out those bacteria which are not being transformed. All right. So that is why we are able to see here no colonies in this agar plate, right? So it is the DNA from heat killed S strain that transforms the R strain when the broth was subjected to DNA's treatment, and the DNA got cleaved and hence resulted in no colonies. You see? But it is just a possibility that it might be DNAs. Further conclusion and further confirmation is given by Hershey and Chase experiment. You see, we know about the Griffith experiment, which followed to discover that the hereditary material in organisms, which followed to discover it. But it was not accepted by all until Hershey and Chase published their experimental results. Their experiments led to an unequivocal proof that DNA is the genetic material. Bacteriophage, that is virus that infect bacteria by the transfer of their genetic material, were the key element of this experiment. Viruses do not have their own mechanism of reproduction, but they depend on the host for reproduction, and once they attach to the host cell, their genetic material is transferred to the host and it gets incorporated within their genetic material, hence to produce further copies of a virus, resulting in the death of the host cells, and those viral copies infect other cells. This is the basic mechanism of how majorly viral diseases work. Okay, here is the basic pathology behind that. But once they in this case, once they attached, I already told you. In this case, they made two sets of experiment. By radioactively labeling either the protein code, which is the second set, they radioactively labeled the protein code with S35. Why S35 was being labeled in the protein? Because many proteins consist of sulfur. You might have heard about it in your biomolecules classes. Okay. And why DNA, why genetic material is being radioactively labeled by 32 phosphorus? Because as we may all know that DNA consists of a phosphate moiety. So it will help in knowing that the DNA is being radioactively labeled. That is why it is radioactively labeled like that. Okay. So what they observe, so here is the entire setup of the experiment. Okay, the bacteriophage was and the bacteria were mixed in a broth and allowed some time to the bacteria, first to infect the bacteria by injecting its genetic material inside it, and then they were subjected to shearing forces in a blender, and after centrifugation these results were obtained. Now I will explain the results. E. coli bacteria which were infected by radioactive DNA, you see here, were the ones who were infected by the radioactive uh, were radioactive. See, E. coli bacteria which were infected by radioactive DNA were radioactive. See, you see this in this pellet, and in this bacteria you see the viral DNA is being incorporated in the bacterial genome. Okay, and the bacterial genome, this pellet is made up of bacteria and it has radioactivity, but the ones who were infected by radioactive proteins were protein viruses. You see here, there was no radioactivity in the genetic material of bacteria. You see, although both were infected by uh, bacteriophage, but in one radioactive labeled DNA was there and in one protein was labeled. So it was concluded that protein is not the genetic material. It is the DNA which is the genetic material. It is the DNA which is getting transformed and getting incorporated into the bacterial genome. Right? I hope this is clear, and it is confirmed that it is not the protein which gets transferred into the bacteria. Proteins never get transferred. It is the genetic material which get transferred, right? So now it was concluded that DNA is the one. It was now finally concluded by Hershey and Chase. So here is a few basics of the nature of and the chemical subunits in DNA and RNA. First is the phosphate group. Second is the ribose and ribose sugars. That is, it has two types. One is the ribose and one is the deoxyribose. You may see here. There is an oxy group, and here is no hydroxyl group. OH, no OH, right? So this type of sugar is present in DNA. That is why it is named as deoxyribonucleic acid. And this is present in RNA, ribonucleic acid. That is why it is named as ribonucleic acid. And you see, here are a few nucleotide bases. Uracil is mainly found in RNA. Cytosine, thymine, adenine, and guanine, they are found in both RNA and DNA. Thymine is not found in RNA. It is present only in DNA. And you see here, this is the 5, and DNA synthesis occurs from 5' to 3' end. It runs from 5' to 3'. Now why I am saying it 5'? Because here you see, I'll take this pointer. Here you see that it is the 5th end of the ribose sugar, or 5th end of the sugar backbone that is free to form bond. And here it is the third carbon of the sugar backbone which is ready to form bond with the phosphate moiety. That is why it runs from 5' to 3'. And to the first carbon, nucleotide bases are attached, and you may see here that adenine and thymine forms double hydrogen bonds, while guanine and cytosine form triple hydrogen bonds. So in the next section, we will be discussing about the basics of the genetic material, that is the DNA and RNA. Therefore, and the central dogma of biology, that is replication, transcription, and translation. And to summarize, we learned about the basic differences in DNA, genes, chromosomes, and alleles, and we reached to the conclusion that how we studied about those experiments, that how we discovered that DNA is the genetic material. Thank you so much, and we'll meet in the next lecture.